Hey there! So now that we have a little bit of DNA structure and replication under our belt, we're going to start to take a look at how DNA actually codes for the traits that we have and the proteins that it produces from its information stored in the nucleic acid code of all of the nitrogen bases. So that's where we're going to go with this one. So we're going to open it up, a little bit of backstory, and then work our way into the actual process of going from the code in the DNA all the way to the protein product. So, genes and proteins, very linked. The idea first came around that there was going to be one gene for one enzyme. And that was first and foremost put together with the work of Beadle and Tatum. And they did their experiments on bread mold, okay, so Neurospora. And what they did was they took the bread mold and they basically blasted them with some x-rays and found that as a result of these mutations to this particular gene when they were using the x-rays, they found that the Neurospora lost its ability for one enzyme, which was to break down part of food for metabolism. And it lost its ability for this enzyme to work. So they showed that there was this gene, and if they mutated the gene with x-rays, it caused this effect on the other end, which was a non-working enzyme. Okay. But what we have to remember here is that not all proteins are enzymes. In fact, proteins play huge roles. They're hormones, they're transport, their receptors, their coats, the proteins wear many hats. So an example is sickle cell anemia, and we've talked about this before. We have our hemoglobin, which is the protein that binds to oxygen in the red blood cell, and when you have a working red blood cell, it looks just like this, okay? So very, very happy red blood cell. Well, when the hemoglobin is affected and isn't structured properly, it gets really sticky, can't bind to oxygen, and as the cells work their way through, they create this sickle shape. So this is a not so happy red blood cell. We also have things like the protein outer coat on a virus, and this was, if you remember, very tantamount to Hershey and Chase's experiment that they did, which told us that, hey, heredity, it's all about DNA. Whee! Okay, so that outer protein coat is basically what creates that shell, or what holds the actual virus shape together. So really what it comes down to is the fact that we have one gene that promotes the formation of a single polypeptide. And if you remember, polypeptide is the name we use for the amino acid chain. It's not an actual functioning protein yet because it hasn't been folded or modified or created into a working protein. It's a more appropriate explanation, but even still, there's still layers of complexity here. We can have one gene code for many different proteins, and that comes down to RNA modification, which we're going to talk about later on. So it's a good statement. It was a great statement for when we had Beadle and Tatum. It was the push. It was the push for the hypothesis behind the number of genes we were going to have at the end of the Human Genome Project. But we were awful surprised when that ended. All right, so the actual genetic code. Let's talk about that. We know 4 nucleotides, okay, in DNA: adenine, guanine, cytosine, thymine. There are 20 different amino acids in all of your proteins, okay? So we can take those 20 amino acids and we can put them in different orders, lengths, and create infinite chains. So that's why we can have so many different proteins. So how do those 4 nucleotides end up coding for such a vast number of proteins? Well, we begin by talking about it in terms of triplets, okay? And that was first suggested, and it turns out that that was right, because if I start talking in triplets, meaning like A, C, or A, T, G, and so on, and talk about DNA in these triplets, I can actually create 64 combinations with 4 bases. Okay, so I can have AAA, I can have AAC, AAG, AAT, you know, and so on. And I can create 64 of those. And each one of these will end up coding for 1 amino acid, which can fill the difference. So the primary protein structure, going back, is all about that sequence of amino acids. So it's these triplet codes that are going to determine what amino acid goes in what order. And we're going to talk about how that works too. So just to show you about that 64 idea, if I only have 1 nucleotide per amino acid, I can only have 4 different amino acids. That doesn't work because I have 20. Okay, so I can't make that work. If I use 2, I can only get 16. I'm closer to 20, but not quite there. But if I use 3, ding, 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 okay, I can have those 64 combinations, which means that I'm going to have multiple triplets coding for the same amino acid, which is a really, really important thing that we're going to get to. So the 3 nucleotide sequence we refer to as a triplet. Here we're going to have some semantics here, okay? This equals 3 bases DNA, okay? That's really important. The DNA in 3's triplet, okay? We'll talk about what the 3 in RNA mean and the 3 in a different form of RNA. So there's different terms we use depending on which molecule we're talking about with the 3 base pairs. So the amino acid code then, you know, basically is degenerate. We have more possible codes than amino acids, and it turns out that we can have, you know, there's up to, I think 6 is the most. You'll have 6 different triplet codes that can code for the same amino acid. And we're going to show you how that works. And luckily for you, scientists have figured out what the code is. So pretty much where we're going to go is how you read the code and how you actually create that amino acid sequence. So where we're going to go is how you read the code and how you create that amino acid sequence. So stay tuned because it's going to be a pretty fun event. So take it easy and we'll see you guys later.